Well, I'd like to just jump into questions and have a variety of people answer. So if you have an answer or multiple people want to answer the same question, I think that would be great. You talked a little bit about this in the case studies, but Hari in New Mexico had a question. She said, please explain the thinking of the IMWG standard of care for starting relapse, like a 25% increase from baseline. She's saying, as far as I can find, there's no data that justifies this approach versus like starting versus waiting. So if you're really considering these patients are high risk, and I've had this question too from people who are looking at it, even at the MRD level. Like if I'm starting to see my numbers go up, whether it's MRD or whether it's my monoclonal protein, why would I wait at all? I mean, why isn't that an indication that I just go for it and just, I need to start a new therapy? My therapy is clearly not working.
There's two reasons why you wouldn't start therapy immediately in everybody. Again, it's a highly customizable disease once you relapse. I agree with, we can have algorithms for people who are newly diagnosed and all that, but after the first relapse, people start splitting off in different ways. So the problem is this: we have limited number of treatments for myeloma. Many people, if you think of all the people in this world who are living with a monoclonal protein in their body, most people don't need any treatment. They all have MGUS. A lot of people have smoldering myeloma. So just having a monoclonal protein show up in your body is not an indication to start treatment. The second patient that we just discussed had a clear-cut kidney dysfunction when she presented. Her body has already told us that the presence of a monoclonal protein, even a small level, is associated with kidney dysfunction. That person cannot wait. I have a patient, for example, with high-risk myeloma who I transplanted, and I have to talk to her every month just to keep her happy. But she's had myeloma, high-risk myeloma, really clear-cut high-risk myeloma, P53 and 414 in 2009, transplanted her in 2009, and relapsed in 2017. Now, the last 3 years, she has not received, her IgA M spike keeps going up little by little by little. It's almost 2 grams now. And it's a slow progression. It is not doubling over even in all these 3 years now. And she does not have any bone disease. I scan her with a whole body CT scan every 6 months just to be sure that nothing is happening. And now with the pandemic, we decided, we were about to treat, and then the pandemic happened. We've now waited another 9 months. Nothing's happening. So I've added 3 years to her life without doing any treatment. So when I used daratumumab and pomalidomide for the next round, her clock starts at that time. So it's 3 years of free time I added to her, but it's a very customizable patient. That I wouldn't do this for the majority of my patients. This is an exception. So that's the thing. Once you're relapsed, it's always a little bit of a different thing with each patient.
So you're looking for like rate and rise almost.
Rate of rise. How likely is this person's myeloma going to damage their body? Many people have what we call smoldering relapse, which means they're back almost in a smoldering myeloma where it's going up little by little by little.
Yeah, okay.
So if we had a curative treatment, so if we find that early CAR T therapy will cure this person, and if that's what the next 5 years brings, yes, we would treat them immediately when the number goes off. Right now, we have to live with the fact that none of our therapies are curative, and every therapy has a limited time span. So if you can, by some crook or means, get it 2 more years later in their lifespan, we're adding those 2 years to the lifespan.
Yeah, that makes a lot of sense. Okay, I haven't thought about it like that before. I have a question also. When you're thinking about all these new immunotherapies, and especially CAR T and potentially the bites, I don't know if they have the same issue or not. But sometimes people say, okay, when you've been on all these different lines of therapy, your immune system your T cells are more exhausted or they're kind of more beat up. So like which drugs specifically are depressing the immune system? And I've had people tell me that Dex is one of the biggest culprits. Are there others that we need to be patients are trying to say, okay, if I'm going to do CAR T, what can I do now to maximize the strength of my immune system while not preventing me in the future from doing that? And they're weighing all those choices all the time. So as you're considering all these lines of therapy, how do you maximize that immune system function?
I think there are, a couple of different issues here, and I think this is a really common question. And it's not clear to me that it's just the drugs. Clearly myeloma itself, has an effect on our immune system. You look at patients at the time of diagnosis, many times their CD4s and CD8s are low, like what we might see in another immunodeficiency disease. So I think it's probably more complicated than we probably know. But it's also clear that over time, with progression of myeloma, that the immune system also changes as well. So there's some kind of interplay between our immune system and the disease. And certainly some of the treatments we give and administer can make that worse. So transplant, for example, we can see suppression of T cells after transplant that can recover over time. And then there are other treatments that are just toxic to T cells. One of the ones that comes to mind for me is bendamustine. Not to pick on bendamustine, but it is just really toxic to lymphocytes. And we use lymphocytes to make CAR T cells and to get a response with bispecific antibodies. So if there's any drug I would probably avoid, it would be bendamustine and others of that class. Any alkylator, nobody gives oral melphalan and prednisone anymore, but that would probably also fall into that category. Mm-hmm. But interested in what others think as well.
Yeah, it's a complicated question. I think at this point we don't have enough data to be saying we should, aside from the ones that Dr. Cowan just mentioned, but certainly in earlier lines of therapy, that we should be avoiding good drugs just because of potential effects later on when CAR T cells are going to be delivered. I think what we know is that if we give really effective anti-myeloma therapy, we can make the immune system better. Yes, there are still going to be residual changes, but ultimately, I think we need to focus on giving best lines of therapy, getting the best responses, and until we have data, especially with moving CAR T up earlier in lines of therapy, until we're at that point right now, I would just say use the best drugs you have, aside from doing things like heavy doses of alkylators.
Mm-hmm. Okay, makes sense.
Well, it's still yet to be proven. One of my interests is potentially using some of the leftover autologous stem cells that are not going to be used for a second transplant. There are a few case reports of patients whom developed severe low blood counts after numerous lines of therapy, were ineligible to go on and receive additional lines of treatment and were at risk for recurrent infections. And giving back those previously collected stem cells sort of winds back the clock a bit, giving and boosting up blood counts so that they can be eligible for more therapy. So that's certainly something that we've tried in a couple of patients at our center and maybe worth considering for you.
That's so interesting. I've heard about like freezing stem cells, but Dr. Hari, you kind of answered some of the questions that I had about that. And I don't know if I can make a public comment about that idea.
Yeah, so so there are two things at play here. One is that why not collect and freeze your T cells when they're good? For example, you recover from an auto transplant and you're on, say, rev maintenance, or even before you start rev maintenance. One of the few cells that come up is T cells. You may have fresh T cells. They might have come up from the newly infused stem cells. Why not collect and freeze them? There is a thought about doing that. There are thoughts about collecting T cells at diagnosis, which are basically antigen exposed, but not functioning in some way. And then if you can redirect them with CAR T or something, it might work better. But what Dr. Coffey just mentioned is the most important thing for patients, for example, who've had multiple therapies, their immune systems are beat up, they don't have a lot of T cells. You could actually just do a transplant at that stage. Even if you don't expect that transplant to give a long sustained myeloma benefit, you could just do it to get some good quality T cells and then do it. The other option, which might again become a future option, is that you may have stored product from before, which is basically your stem cell product that usually has plenty of T cells in there, and you might be able to use that to make CAR T cells. People are thinking about many different approaches. But like Dr. Cowan just mentioned, bendamustine is a terrible drug when it comes to, I have certainly been burnt by patients who I wanted to do CAR T, where their T cells were not even detectable after bendamustine, and I had to bail out on that plan. High-dose dexamethasone. We don't use super high-dose dexamethasone these days, but that can definitely be a very powerful lympholytic drug. So, it's something that patients will need to be more and more concerned about. going forward. But there's one myth again that's out there, that if you do an auto transplant, then you're never going to get CAR T cells, or if you do an allo transplant, you're never going to both Dr. Cohen's group and us, we have given CAR T cells to many patients who've had an allo transplant, and actually the results are very good. I would argue that they're even better than the patients who've had auto CAR T. Yeah, but these are allo derived CAR T cells collected from the patient themselves.
Interesting. That's so it's this whole new world of things, right?
Yeah.
Okay, I'm gonna try to be a little faster with the questions. Seville is asking, what can we do to avoid getting secondary cancers like AML? Not a fun question to answer.
Very difficult question to answer. Sarah's probably the most qualified.
So first of all, you have to understand that just having myeloma itself is associated with an increased risk of myeloid malignancies. So there's nothing you can do to avoid it from a treatment perspective. I mean, that there's a built-in risk. It's low, but there's an inherent risk. And so I explain that to patients when I'm giving the Revlimid post-transplant maintenance spiel, that look, that risk exists just because your body's produced myeloma. The risk exists because you've gone through a high-dose melphalan transplant, and then with lenalidomide maintenance, that risk goes up a little bit. And then I talk about relative risks in terms of, the elephant in the room still being the myeloma, but that real but small risk of secondary primary malignancies. For me, certainly if there was somebody who had signs of MDS or something that looked like emerging MDS heading into a transplant, that would make me nervous, and there'd be long discussions with the transplanters about whether or not that would be appropriate to give high-dose melphalan and about whether it'd be appropriate to give lenalidomide maintenance. But otherwise, on the flip side, if there's never been any evidence of inherent increased risk of MDS or AML aside from having myeloma going through a transplant, I monitor patients on a monthly basis, which I know many people don't. And part of why I do that is A, to pick up those aggressive relapses, but B, to pay attention to blood counts. That's how I've picked up, a handful of secondary AMLs and secondary ALLs, is because people have been on their maintenance, chronic doses, stable counts, and something has changed. And I have a low threshold for doing a bone marrow biopsy. So for me, vigilance is a big part of it.
Good point. Okay, we have a live case study with Greg. He was just barely diagnosed in October of this year. Anemia with fractures with a 414, gain of 1Q, gain of MYC, CCND1, that's 1114, right? MAF is 1416 and TP53 in a large percentage of cells. So with this is, does somebody want to talk about double hit or triple hit? Or you may have already talked about that this morning, Dr. Holstein, but like for an ultra high risk person like this, what do you suggest?
So I guess I was a little bit confused by that rattling off of abnormalities. Were those all translocations?
So 414 in 90
okay.
Gain of 1Q.
Okay.
With like 90% gain of MYC, CCND1, MAF, and TP53 in the 90s percentages of cells. So,
okay. So yeah, the double hit, triple hit comes from lymphoma terminology that some people are trying to apply to myeloma. I kind of tended to have used the high risk, ultra high risk classification that the myeloma 11 uses. Some of the data I showed in my talk was one high risk feature is bad, more than one high risk feature is even worse, and that's why we refer to it as ultra high risk. And I think all the other panelists have already spoken to our approaches in terms of thinking about what we do for induction and whether or not we do 1 versus 2 transplants or transplant allo or clinical trial. But clearly the data show that having more than one high risk abnormality is worse than just having one.
Yeah. Okay. Vicky's asking, do you all see a role for CRISPR in myeloma research? If somebody wants to give just a quick overview what that is too.
CRISPR is basically a tool that was, discovered, quite recently actually, that allows for editing of genes. And so I think, how could it be used? Well, I gave an example of gene editing. They use a different system called TALEN in this study of SLAMF7 CAR T, allo CAR T. And the goal there is to edit out the gene that could result in graft versus host disease, the TCR alpha. So, I mean, I think we're already seeing examples of it being used. I mean, there are certainly, concerns and risks with gene editing, off-target effects and unintended consequences and things, we can't predict when we're, messing whole scale with the genome. But, so I think it has promise, but we haven't, it hasn't really worked its way into, prime time or FDA approved therapies at this point.
Super early, it seems, for everybody, right?
There is one example phase 1 study that was reported at ASH last year from the group at the University of Pennsylvania. And what they did was they used the CRISPR gene editing technology to introduce a T-cell receptor that specifically recognizes an antigen called NY-ESO-1 on the surface of myeloma cells that are being presented by what's called the HLA molecule. And so this is one of the first examples of where CRISPR and myeloma have met. But it was a small study, too early to say whether or not this is going to change practice. But all eyes are on this technology for other applications in health and disease, including multiple myeloma. And I think we're all pretty excited about its potential.
And we do have a phase 1 trial that's we are part of that trial, where we use off-the-shelf CRISPR-edited bcma targeted CAR T, and I think some of you also might be on.
Interesting. Okay, great. Here's a question I don't see very often. So Chris has MS and multiple myeloma, well, smoldering multiple myeloma, and is like pursuing treatment. Essentially, is there anything that you've heard in terms of connections, either with MS or other diseases on a regular basis? I know secondary cancers are kind of, after the fact, but is this just an unusual anomaly?
I mean, I think I also have a patient with SMM and MS, and I think there's not data that I'm aware of that supports a connection between these two diseases, unless others have seen something. Where at least in my patient, we've been trying to figure out is how to get the patient to autologous stem cell transplant. As many of you may not know, that autologous transplants are also utilized for multiple sclerosis, can be quite effective for the severe forms of MS.
Wow,
but currently it's very limited in terms of availability. I think at our center, you have to participate on a study to do that, and having smoldering myeloma would preclude enrollment on a study. But it is interesting, and it's timely because I've been thinking about it also recently.
Right. And the only difference is that when you do the MS transplant, it's a CD34 selected transplant as opposed to a traditional transplant. But that would still work for myeloma because
Wow, so that's a thing to consider. Interesting. I've never heard that.
There are associations between autoimmune diseases and monoclonal disorders like multiple myeloma. MS is an autoimmune disorder. It's possible the two could be interrelated, but I don't see enough autoimmune disease in my practice to say that anyone with an autoimmune disease is going to get a plasma cell disorder. So there's probably environmental and genetic factors that increase this individual's risk of having both.
Mm-hmm.
And I would say in general, we worry more about B-cell malignancies and plasma cell malignancies with a lot of these autoimmune disorders, either because of the disorder itself or the treatments that they get. In my own practice, I've had a variety of patients with other autoimmune disorders, and sometimes there's been some degree of anxiety about whether or not any of the anti-myeloma therapies might exacerbate their underlying autoimmune disorder, and that's really never been the case. And we've always really been able to give standard of care myeloma treatment, including transplant, without difficulty.
Mm-hmm. Okay, interesting. I've never heard of that connection before. So Alyssa has a question. Can you address Nilaro as an alternative to KRD for high-risk disease, I guess, or VRD? I mean, you all talked extensively about VRD versus KRD. How about using ixazomib?
I would only consider it in very select patients where performance status was really an issue. I think out of the 3 proteasome inhibitors that we currently have, I consider, at least the way we currently dose Ninlaro, to be the least potent. There, you know, in the relapse refractory setting, there is benefit for Ninlaro with Lendex in high risk. But at least in the transplant ineligible setting for newly diagnosed, there really wasn't much benefit for adding Ninlaro to the backbone of Lendex for newly diagnosed. So it would certainly not be my go-to proteasome inhibitor for a younger transplant eligible patient. I'd prefer to use either bortezomib or carfilzomib.
Mm-hmm.
I agree.
I
agree.
It's one of those drugs where I've seen more misses than hits. I mean, I've tried it a lot, and I think I've been somewhat disappointed. So.
Okay, Lonnie has a really good question about using Pomalyst instead of Revlimid for maintenance therapy. I've heard myeloma experts say Pomalyst is a little more potent. So why not use something like that if you have high risk features?
We're lacking data. There's just very little data for POM maintenance post-transplant at this point, and it would have been a great study to do for Len versus POM as a randomized study, for example, and even in just high-risk patients. I think just because of the way that these drugs are being developed, that we're going to kind of skip over Pomalyst and start going into cell mods for maintenance instead of lenalidomide.
Yeah, I think that's interesting. It sounds like more potent and lower side effects.
So far, that's what it seems, yeah.
Interesting. Someone's asking you all have mentioned tandem transplants. What percentage of your high risk patients end up getting a tandem transplant, and how do you determine, is there any data that shows actually who does better with this? Like what genetic features might do better?
We actually don't know which genetic features. The problem is we don't have a high-risk specific, we don't have too many high-risk specific studies. So the studies are basically high-risk subset of large studies. And then everybody with high-risk features is put in that high-risk subset or a big basket. And then when you get down to the people with 17p versus people with 414 and the people with 414 and 17p together and all that, it comes to tiny numbers from which we cannot draw robust conclusions. So that's the problem of looking at data. We don't have any data to guide us. What we do know that there is a signal that tandem transplant seems to benefit patients with high risk more than the patients who are at standard risk, in so much so that standard risk patients we can't actually show a benefit anymore. High risk, at least in the intent to treat trial of Staminid, showed that. In Europe, it's still quite extensively used. And then the only other piece of data that we can go by is the people who have achieved a deep remission from a single transplant. At the end of that single transplant, if you're in a really good remission, a second additional transplant may not be worth the effort. So that's the one fork in the road where you say, I'm high risk, I had my first transplant, I'm in a stringent CR with very little disease on board. Maybe I don't need to do a second tandem transplant. I would estimate that I offer high-risk patients a tandem transplant, but then I end up doing it in only about 15% of cases so far.
Interesting. Okay. Question about CAR T clinical trials. Many of the trials from Darlene, many of the trials I've seen seem to roll out people who have, I think she's saying CKD, so chronic kidney disease. Is that what she's saying? Especially with CAR T. Is this related to study design, or does the kidney issues make patients ineligible for these treatments in the future?
Yeah, I wouldn't say it's so much that the kidney disease makes, the CAR T not work. It's really just, trying to, tailor the inclusion criteria of these studies to, select unfortunately the companies and investigators want to have the patient population on study with the least comorbidities. And, whether that's fair or not, I think is a debate, beyond this. And I think, this is where I think looking at investigator-initiated trials at different centers is more useful. Then there's more, sometimes investigators have more leeway on how they write the inclusion criteria. And what unfortunately this also means is this, should there be a CAR T product approved for myeloma, insurance companies will likely, approve, use of the product based on what the eligibility criteria were for the study. This is what we saw with Yescarta and Kymriah, which are CD19 CAR T cells for lymphoma, is that's how they would say whether they were going to pay for it or not. And fair or not, it's how the system is currently built. So I always think it's important to really look at eligibility criteria and think, like, what are we trying to do here? We don't want to unfairly exclude patients, and we saw this for years with HIV, where every study would exclude HIV, and I think there's been a push recently to try to make that more clear. We want HIV is okay as long as the viral load is undetectable and so on. And so, but the CKD, especially on dialysis, becomes an issue, and you have to find studies that are for patients on dialysis.
So kind of along that point, when you're talking about upcoming approvals for the Bluebird BioCellgene CAR T and for the Janssen, which hopefully will come next year, what kind of, those studies have inclusion criteria, like you have to have prior DARA or prior CD38 or you have to be refractory to lenalidomide and things like that. What do you expect when those are commercially approved? Similar? I mean, you still have to have those as a patient, or can you just say, I've had one prior line of therapy and now I want this?
I don't think that's the way it's going to work, since these therapies are so incredibly expensive. So unless somebody's paying out of pocket, which I think would be a very small percentage of people who could do that, insurance companies are going to have to approve it, and they're going to look at the label, which is going to be based, as just was discussed, on the clinical trials that led to approval. It's frustrating because we know that can be a big disconnect between the patients in our real world clinics and those who were put on the study. And it was certainly frustrating as an investigator to have to keep having screen failure after screen failure after screen failure for things that, ultimately I thought were not that important, but that's the way the study was written. So because these products are so expensive, I think insurance companies are going to be pretty stringent about following the label.
Yeah, but wouldn't earlier use? I mean, that I guess that's the issue patients are going to have too.
That's why all those clinical trials are being done. So Dr. Hari, I think, mentioned a few of them. So those studies moving it up are being done, but we can't just kind of, be lawless and just start doing whatever we want to do unless magic money comes from somewhere.
Yeah, I understand. But it's frustrating nonetheless, I think, for patients who are looking at CAR T as okay, it's working great for these patients with 12 plus lines of prior therapies, and let's move it up faster and see if we, all the things we've talked about today. Okay, Wendy has a question about her husband who had a stem cell transplant and had CAR T, has high risk features, deletion 17 and 14, 16, but has relapsed, or would be relapsing. So in that situation, do you go for try to do another CAR T, like with I can never get this right, GPRC5D target, like a new target? Or do you do a BiTE, with that target, or go for the antibody drug? You probably wouldn't want to do the same BCMA target, right? Or would you?
Again, we actually have no data on whether we can have a second go at with a different agent. I have to say, I have patients who failed a BiTE and then had a CAR T and responded. Don't have too many people going the other way yet, but we will generate those data. There's a published report of somebody who failed CAR T and then went on to have Blenrep or belantamab and had a pretty good response. And I'm sure Andrew has a lot of experience in that too.
Yeah, I've seen both. I mean, we've had patients on our studies who've had a BCMA BiTE, no response, and BCMA CAR T, and it works beautifully. I mean, it doesn't make a lot of sense. There must be some reason for this. In CAR T, and even though we think of these interchangeably, CAR T BiTEs, they're not exactly the same. I mean, with a CAR T, you have an engineered protein, on the surface of a plasma, on the surface of the T cell. So that's different than giving an antibody that attaches the two. They're not, exactly the same treatment, although, mechanistically they're similar. I think, what seems very provocative to me was this report from ASH where someone actually has a BCMA deletion. So that's on 16p, I think, is where BCMA is. And
Yeah.
so, and then an inactivating mutation. And so, if you think about it, all it takes is one myeloma cell. That's it. And myeloma is heterogeneous. So that's why I really feel, I firmly feel that, we need like 2 or 3 targets to treat patients,
Or
because
dual targeting, right? Yeah.
Exactly.
Mhm.
Yes.
That makes
And
a
there
lot.
was,
I have an
Go ahead.
unusual case of a patient who went to China, received a BCMA CD38 dual CAR T cell, had no response, came to our center, got a BCMA CAR T cell, and had a durable response. So there are very unusual examples. And I think if you fail one CAR T cell, it doesn't mean you will fail them all. Or if you've had a good response to one, you may not have a good response to the same one again in the future. This is just too much to know. But I do think doing testing to see, are you still expressing the CAR T cell protein before going on to that therapy would be a consideration.
and there was some intriguing data from last year's ASH out of China, where they had a couple of patients who had previously had a BCMA CAR, but it was a mouse construct. And then they went on to the clinical trial that they were reporting, which was a human BCMA construct, and had responses after they had progressed on the first BCMA product. So we just need to see. The problem has been so far with many of these studies that is that they exclude, prior BCMA therapy, or probably moving forward, they might exclude prior CAR therapy. So we just have to be flexible in study designs.
Yeah, I appreciate that. I think about the Hutch, that you've gone out of your way, I think, to not exclude people with prior.
Yeah, that's why I need center-based CAR Ts more than pharma company-based CAR Ts. I know that we have a lot of pharma support, but to be innovative and flexible and to have benefit to maximum patients without really looking at FDA approval as the only goal, we need to have centers also do their own stuff.
Yeah, and potentially lower cost, right?
Yeah,
and
absolutely.
improve speed. Yeah. So it's not 4 to 6 weeks, it's shorter. Yeah. Okay, Bonnie's asking, can you comment on light chain disease and high-risk cytogenetics? So light chain myeloma only. Is it the same? Do you just deal with that in the same way? And then sometimes the clinical trials exclude these people and non-secretory people, which is a little frustrating for people who are down the road quite a bit and have developed non-secretory myeloma. Do you see any changes happening in the future for that?
So I guess I'll take a first stab. I don't think of light chain myeloma with high risk disease as any different than IgG kappa myeloma with high risk disease, or IgA lambda. Most of the time these days, you can have measurable, there's criteria for what measurable disease is from a light chain perspective. So I can't recall the last time I've seen a study designed where they excluded light chain only disease, but absolutely the patients who don't meet criteria for measurable disease, so they're kind of oligosecretory or they're truly non-secretory, that's where it's oftentimes very frustrating because of lack of clinical trial options.
Mm-hmm. Okay. I know Adrian had a question about BRAF mutations. So there are targeted inhibitors, for these types of mutations. They are considered more high risk, like these NRAS and KRAS mutations and BRAF mutations. How do you handle that? Because if there are hundreds or thousands potentially of mutations in myeloma, is taking a single targeted inhibitor going to do anything?
Unfortunately, the identification of a BRAF mutation, for example, in multiple myeloma has not proven to be enough to be able to give a single agent to target that mutation and expect a complete remission. And the reason for this is partly because multiple myeloma is so heterogeneous. It's not a single mutation driving the disease. And so while you will knock out the BRAF positive myeloma cells likely in the cells, in the body, there's going to be others that may not have that BRAF mutation that continue to grow. As Dr. Cowan indicated, it just takes one mutant variant that can kind of take over the population of cells. So I think multi-drugs are necessary. It would make sense to consider using a targeted therapy in combination with other drugs that have maybe more non-specific effects, so that you can eliminate cells that don't have that target mutation.
Mm-hmm.
And I think, did you talk today, Dr. Cowan, about our study screening patients?
I did not, but feel free to go through
Yeah.
it.
So at the SCCA, we are doing a novel clinical trial where we'll take a biopsy of the bone marrow, and then we will take cancer cells from the bone marrow, and we will plate them in a dish. And then we will put into each well of that dish 170 different drugs. And then we will culture that in the lab, and we will see which drugs may potentially be more effective in the lab. And then we could provide that list to the treating physician, and then they can choose from that list which drug to give the patient. So that's an example of trying to address hard cases where the common myeloma therapy is no longer effective, and you need to try and use some other cancer-related treatments, but don't know which one to pick.
Yeah, I love that effort. I'm hearing more and more about that. I know Dr. Rodriguez at Wake Forest is using organoids to kind of do the same thing in combination. So I love that you're doing that. That's fantastic. Somebody's asking just a general question about enrolling in clinical trials in the United States. Do you have to be a U.S. resident to enroll in trials in the U.S.?
Generally,
No.
you have to live close to where the trial activity is happening. So unless you can stay in the United States, it's not possible. And we have certainly got patients who've come from abroad and participated in a transplant or a CAR T trial, and then have gone back to their home country. That's definitely happened. I'm sure Andrew and Sarah have the same experience.
Yeah, we have as well. We've had patients from Australia, from Canada. I think the only issue is that, paying for the costs is the big problem. And so, it would be unlikely to have a governmental healthcare insurance agency pay for a study outside of the country of residence. So a lot of these patients have had to pay out of pocket, which to me is unimaginable, but it's, this shows the efforts to which we will go to try to, get the best therapy.
Exactly.
Okay, I'm going to ask two more questions. George is asking, so since everybody mentioned reusing these stem cells, George said 3 years after transplant, like he decided not to pay for the storage anymore for his original stem cell collection. Like, should he rethink that decision and figure that out so that he has something on hand?
3 years in remission? Yes, definitely. The longer you're in remission, the more valuable these stem cells are.
I agree.
But that's the way it is.
I agree.
Okay, so we should keep those stored. Okay, that's good to know. And then there were a lot of questions about 1Q gain, and I know that there is an inhibitor. I can't remember the name of it. I apologize, but that there's an inhibitor is it MCL1, an MCL1 inhibitor. Have you seen it to be effective at all in the 1Q gain? If people acquire it, the 1Q gain afterwards is, I don't know, what are you learning? We didn't used to think 1Q gain was high risk, but now I think the consensus is from you that it is, right?
I think the context is important, but I'll be honest and say I don't know any specific data about 1Q and the MCL1 inhibitor. Perhaps my colleagues do.
We have done 2 trials with MCL1 inhibitors. They were not limited to 1Q gain patients, but both of those studies did not move forward after phase 1. So that tells us something. One was because of excess toxicity, and the other one was no efficacy in myeloma. But there are still companies trying to develop MCL1 inhibitors. And the 1Q gain by itself, Dr. Holstein is absolutely right. It depends on the context. In the setting of other mutations, it definitely adds, it synergizes with the badness.
The other thing I think is important is also what's going on with the 1Q? Is there one copy? Is there 4 or 5 copies? Sometimes people differentiate between a duplication and then an amplification. And so the amplification seems from some studies to have, portend higher risk than just a simple duplication. And the reason why I think, just to put some context about the MCL1, because MCL1, the gene for that is found on chromosome 1. So, there's probably at least some, preclinical data to support that maybe, higher, expression of MCL1 could be deleterious in myeloma, and therefore an MCL1 inhibitor could be effective. But as Dr. Hari mentioned, those studies have not really been fruitful so far. Although we've learned never to give up on drugs, right?
Yes.
Okay, well, I just want to thank you all for joining today. Each of your presentations were outstanding. I think this conversation is so important because you're answering such specific patient questions. Thank you for taking your Saturday. Thank you. And we just appreciate everything you're doing for myeloma patients. What's happening and what you're all working on is just truly stunning. It's so stunning to me as a patient. It gives me a lot of hope. So I agree. We have reason to have a lot of optimism. And thank you.